resolution molecular structures corresponds to lower numbers. This is because resolution refers to the distance between two things at which you can still tell that they're two separate things. So if my fingers are really close together, it's harder to tell if they're how many fingers I have holding up, as opposed to if they were far apart. And so the closer together two things can be, what you can still make out that they're two separate things, is the resolution. And so a smaller number is better because you can still tell that they're apart. So you can also think of it as like point sources or like stars in the sky, not being able to tell if it's two stars or one star. So resolution is super duper important in things like X-ray crystallography, where we're trying to figure out the atomic structure of things like proteins. So basically their molecular architecture at the atomic level. So all the individual carbons, nitrogens, oxygens, that sort of thing, and where they're placed in 3D space. So if we have high resolution data, we can tell those apart really well. We'll go way more into the details, but basically with X-ray crystallography, what we're doing is we're shooting X-rays, so like really energetic light waves at molecules and crystals. And basically what's going to happen is those X-rays are going to interact with the molecules, more specifically with the electrons in the molecules, and those electrons are going to scatter the X-rays, and then the scattered waves are going to interact in ways that are going to give you this pattern of spots called the diffraction pattern. You work backwards from that pattern, you'll get the map showing you like the electron density map. So basically showing you the positions of the electrons. When you see one of those like sticky models, basically those are showing you like the atomic position. So basically they're showing you like where the center of the atoms are, whereas the electrons are kind of like moving around, like they're whizzing around those atomic nuclei, which is like you're modeling basically the nuclei, but what you're seeing is the electron density. And so this electron density is kind of like this map you get can be like fuzzier or not fuzzier, and then you're building this like model into this map. The fuzzier this map is, the lower the resolution, the harder it is to accurately place the structure into that map. Whereas if you have really high resolution data, your map is going to be really crisp, and it's easy to pinpoint exactly where to place those various atoms. So kind of like how two people might be able to look at two fingers, and one person might be able to tell that I was holding up two fingers, and one person might only think I was holding up one finger, one person might think I was holding up three fingers or whatever, because they're guessing. But anyway, instead of having like our crispness of our eyesight, we're talking about like the crispness of our data kind of is going to come from the electron density map. And the fuzzier the map, the poorer the resolution and the higher the numbers, because we can't tell apart that like those two things were kind of merged together. Whereas if we have a high resolution, so low numbers, then we can tell those two things are apart. Our map is less fuzzy. We can more accurately pinpoint exactly where things are supposed to be in it. So you can think of basically the resolution of a structure is kind of like the crispness of our eyesight type of things. So when we have higher resolution data, so we're talking lower numbers, we're going to be able to tell apart the positions of every atom. Whereas at lower resolution, so higher numbers, we're basically going to have to kind of do our best guess to fit the atoms into the structure, but we can't really tell very well where the things are actually going, where the different atoms are within this like fuzzy region. So typically the resolutions are reported in angstrom, and so it has this like big A with this dot. And it's a distance of 0.1 nanometers. So that's what happens when you work with like nanomolar more than nanometers. But anyway, so a nanometer is a billionth of a meter. And so we're talking really, really small distances. If we look at interbond distances, so the interbond distance is basically, it's going to depend on how big the atoms are, like how big their atomic radii are, and then like the bond strength. And so when you have like a covalent bonds, when you have these like atoms linking up to form molecules, basically then they kind of like merge part of their electron clouds, and so they're going to get closer together. If you have bigger
atoms, though, they're going to have a longer bond than if you have smaller atoms. And so with hydrogen, we have a really small atom, and so those bonds are going to be shorter. And hydrogen carbon bonds are about like 1.1-ish angstrom. Carbon bonds, because those carbons are bigger, you're going to be getting closer to like 1.5-ish. And so when we talk about like atomic resolution structures, typically we're referring to around like 1.2, where we can make out like all the individual atoms. So like the carbon hydrogens one's like the shortest. And so if we can see all of those, then it's like true atomic resolution. But typically with like X-ray structures, we're talking more around the 2-ish range. Depends. Smaller structures, you often get like a higher resolution when you're talking about like big complexes where you have a bunch of different proteins and maybe also some nucleic acids, so like RNA or DNA. The resolution tends to be lower overall, because you have a lot of different coordination of different molecules that you have to deal with. But you can still get really important information, even if you can't get the super duper high resolution data.
So with like proteins, we can have with high resolution data, not only can we tell where the various amino acids, so where the letters are, but also like what direction their side chains, so their unique parts are sticking out at. Whereas with low resolution data, we kind of just have to more guess like where those amino acids are, and basically a lot of times we can only see like the backbone, so we can't really see and model the side chains very well. Sometimes we can't even see a region, and so we have to leave it out of our model. And so let me tell you more about what I mean by all of this and where resolution comes from, and how you can assess the resolution of a structure and realize that the resolution is kind of, the reported resolution is kind of like an overall value. But if you want to get into like individual regions, some regions might have more or less like better quality data depending on how flexible a region is and how like move aroundy it is. And we can look at something called the B factor as well, to help get an idea of how like reliable the data is for a region. Not that it's like the researcher's fault or anything, it's just that certain regions of a protein might be more flexible in moving around, and this is going to make it harder to get super duper tight data for that region.
So a basic overview of X-ray crystallography is that we take some molecule that we want to figure out the structure of. This is often a protein, sometimes like a protein and some nucleic acids, so like DNA or RNA, sometimes nucleic acids alone, but we'll talk in terms of proteins. And so proteins are made up of amino acids. So we talked a lot about amino acids and 20 days of amino acids. And so there's 20 different ones, and they have all these, they have this generic backbone part, and then these unique side chains or R groups that stick off and give them unique properties that influence how the protein folds and functions. But all of these different things are made up of atoms, which are made up of smaller parts. So these subatomic particles. So these have at their core, they have this dense atomic nucleus, which has positively charged protons and neutral neutrons. And then whizzing around all of those is a cloud of negatively charged electrons. So atoms actually share pairs of these electrons to form bonds. And it's these electrons that are actually going to be interacting with the X-rays. So when we get the information from X-ray crystallography, what we're really going to get is information about the electron's positions. And because these electrons are kind of like in these fuzzy clouds, we're then going to have to build in the atomic structure. So basically like the model where we put in basically the positions of the various atoms, so like those sticks and things that you see in the structures. But in order to do this, what we're going to have to do is we're going to have to use this kind of roundabout method to get information. And so we have to get the proteins to form these crystallize. And so basically this means we have to take these proteins that are floating around in solution, and so they're really happy, they're hanging out with water. We have to make them give up some of their water bonds to form crystal bonds. And we get them to do this in this orderly manner, or well, we don't like, and they do this in this orderly manner. And when they do this in this orderly manner, we get what we call a crystal. And so basically in a crystal, it's kind of like a brick wall, where each of the individual bricks is like a unit cell. And I'm not going to go into it too much here, but basically you have this repeating pattern throughout your crystal where all of the molecules are oriented in the same way, and you get this pretty crystal. When you shoot X-rays at the crystal, what's going to happen is that these X-rays are going to interact with the electrons. And what they're going to do is the electrons are kind of going to scatter the waves. These scattered waves are then going to interact with one another and either build up, so constructively interfere, or cancel out, destructively interfere. And because of the repeating structure of the crystal, basically they're going to cancel each other out everywhere except for very specific conditions. And so when those specific conditions are met, then you get a spot. And then we can work backwards from these spots to try with this like Fourier transform to get this diffraction, go from this diffraction pattern, so from this pattern of spots on our detector, to an electron density map showing us where the electrons are. And then we can build an atomic model into that map. Because you have like this regularly repeating pattern in the crystal, it makes it so that we can make this kind of like simplification where we can use this, we can kind of visualize slicing our crystal into these planes called Bragg planes, which are places where those conditions for the complete constructive interference are met. And because of the nature of the crystal, everywhere else is going to cancel out, except for in this places where the Bragg's conditions are met, which we can imagine as these planes that we're slicing our crystal through. When we have this, and that the X-rays are bouncing off of, when we have this, this concept of the planes like slicing through the crystal, now we can think about how this relates to our idea of resolution. So with resolution, we're asking the question, how close can objects be and you can still tell them apart? So say you have these 4 dots and you're trying to see if you can tell them apart. Well, if you have it, basically, if you were to slice these in different ways and then see, can you tell them apart? And so if you were to, the thinner the slicing is, the more information you're going to get. And so basically, if you think that you're basically averaging what's in each of these different parts. And if you slice them into more parts, you're going to be able to tell better apart what's where in here. And of course, the direction you're slicing is going to matter too. So you're going to have to combine information from different slicing directions. And this is what we do in X-ray crystallography. Basically, we can imagine having our crystals as being like sliced by these individual planes that X-rays are bouncing off of. And this is where our information is going to come from. The thinner the slicing of the planes, the higher the resolution of the data that we're going to get. The problem is that the higher that thinner slicing is going to correspond to, so the thinner slicing that's corresponding to the higher resolution data, it's basically going to be the hardest to collect. It's going to be the weakest data. So when we collect this data, this isn't actually how it works, but you can think of the X-rays as kind of bouncing off of these invisible planes. And the way that all this geometry works out, and so the spots that you're going to get are going to correspond to these like families of planes that you're slicing your crystal through. And so the planes that have the closer spacing are going to give you the spots that are farther away, and the planes that have the bigger spacing are going to give you those dots that are closer to the center. So your higher resolution data is going to be farther out, and your lower resolution data is going to be closer in. That highest resolution data is going to be at the farthest away in your diffraction pattern. So it's going to be like out on the very edge, and it's going to be like the weakest data. So you get this really strong data for this low resolution information, and this low resolution information is really important, especially when you're trying to figure out like your phasing and trying to figure out like your space group and all of this various stuff. But it's this high resolution data that has that really crucial information, that like high resolution information, and this data is going to be like the weakest and hardest to collect, and therefore and it's going to be the furthest away from like the bullseye in this pattern. Because of this, we're going to there's going to be like a cutoff point at which we can't really say anything from the highest resolution data. So even if our detector was able to detect that there were spots there, we wouldn't be able to really do anything with the information because it was too basically like kind of like unreliable. And so you might see it in like one image but not another, and it might be really super duper faint and you can't really tell it apart from the noise. So basically you have to cut off the resolution where the data starts becoming like useless. And so you have this cutoff, this resolution cutoff, and it's typically like determined by the software and stuff that you're using. And there's various different ways that people have to determine their cutoffs. But basically then you have this higher resolution, you have your highest resolution is going to be limited by your experimental data and by where that cutoff point is. So if you cut it off further away, then we have higher resolution. If you cut it off closer, you have lower resolution. But it's not like we're just willy-nilly saying, okay, we're going to just like cut it off at like the super, super duper furthest, because we have to keep, we're basically only want to use information that's useful. And so if there's no useful information out here, then you don't use that information because it'll just like mess your structure back, like all of your other stuff up. And so so typically what's actually reported as well is like you report the data for like the highest resolution, like shell or whatever, and then also for the entire data set, because this high resolution data is less like reliable. And so it also helps make sure that people aren't just like setting their resolution like way too high, saying it's way too high when that data is really basically useless at that time. It's important to note that the resolution like cutoff or whatever is going to be for the entire crystal. And so basically this diffraction pattern is coming because of the atomic makeup throughout the entire molecule. And so we're getting like you can't just take one of these spots by itself and figure out really anything. You need this entire pattern, and this entire pattern is going to be giving you information about the entire molecule, because it's how all of those molecules, atoms, and their electrons, how they interact with the X-rays to create these patterns. So you're getting information about the entire molecule, and when you're getting in this diffraction pattern. And so the way you set the resolution limit, you're losing information about all of the regions when you don't have the high resolution data. But that doesn't mean that there aren't regions where the data is like fuzzier, or and so there are, and we'll look at that later. Those regions often have what we call like a high B factor, which is sometimes called like a displacement factor or a temperature factor. I also just wanted to mention that there's kind of some like, it's harder to estimate a resolution for like CryoEM, so cryogenic electron microscopy, where basically what you're doing is you're taking a bunch of pictures of these molecules that are in this like really thin layer of vitreous ice. So basically it's kind of like glass, and then these molecules are stuck in space, and then you take a bunch of pictures and you average the information together, and then you can figure out like a structure. because you don't have like that diffraction pattern where you're like setting this resolution limit or whatever, this is going to, it's harder to like estimate the actual resolution. And so it's hard to compare directly between like the reported resolutions from cryo-EM structures and X-ray crystallography structures. And so I'm not really going to go into that, but just know that you can't just like easily directly compare between the reported resolutions for cryo-EM and for X-ray crystallography. And there's some controversy and stuff over how to report your resolution, determine and report your estimated resolution for cryo-EM that I'm not going to get into, but I just wanted you to be aware. So if you had like a super, super duper perfect, well-behaving, immaculate crystal, the resolution would be limited by basically your detector. And so how, like, if you can imagine, you could have even higher resolution data that's just like it's not going to hit the detector because it would be like bouncing off like angles that wouldn't even hit the detector. But in reality, we're usually limited by the protein, by the crystal. And so you can imagine there are different things that could limit the resolution. One would be if there are like defects in the crystal type of thing. So with the crystal, everything has to be very orderly. It's like the definition of a crystal or whatever. But you can imagine if there's like one of these molecules was slightly off or something, or you could get kind of like regions of the crystal where you have a slightly different lattice, like, so like part of it's like this and then you have like a part of like that or whatever. And so basically any of those things are going to kind of blurry up the data. So remember that our data, our resolution is coming from like all of the various, all of the molecules are contributing. So basically there's like, you can have defects in your crystals that can help lower the resolution, so help make it worse. But you can also have things like that are kind of like inherent to your protein and nothing about like the crystallization itself. So some regions of proteins are more flexible than other regions of proteins. And so this has to do with like what amino acids they have, various things like where in the protein they're located. So things on the surface tend to be more flexible than things like inside. And so basically, if there's a region of a protein that's more flexible, it's going to kind of, you can kind of think of, because that region is more flexible, more dynamic, the signal that you get from it is kind of going to be blurrier and kind of, it can even like kind of like cancel out. And so you can't really make out the region. So the higher like if it's in different places, kind of in the different, if like all the molecules have it in like a slightly different position or that sort of thing, then you have a higher like displacement. So sometimes this is called like a temperature factor, because like temperature, energy, whatever, so you can think of it as kind of being dynamic. But displacement factor is like more like technically accurate, because it could just be like stuck in different positions instead of like moving between them or whatever. But then you have things like occupancy. I'm not going to go into that. But anyway, so basically there's this thing called B factor, and the higher the B factor, the less certain we are about the position of that actual thing. When we look at the crystal structures, when we look at the models where we just see those sticks, then we can't tell that like this, unless we like color it by B factor. But if we go and we look at the actual electron density map, then we can see that those regions tend to be like fuzzier and harder to make out where things like the different side chains are. So I want to show you an example of how you can look at like the electron density and show you what I mean by those various things like B factor. So an example, I want to show you this like GMP reductase, the structure of this molecule I was telling you about yesterday, that does this conversion of guanosine monophosphate to inosine monophosphate, which can then be converted to AMP or GMP. And you don't really need to know more about that, but it's kind of cool. But anyway, I thought I would use the structure of this since we had already kind of seen it yesterday. And I was looking at it. So here's its structure in the protein data bank. So 2BLE, if you want to look it up. So you can see that this structure has a resolution of 1.9 angstroms. But I also want to show you how we can see various regions that have like a higher B factor, how we can't even see the regions. So you can see that there are some like unmodeled regions where basically electron density wasn't strong enough to build the model into. Or some of them were like partially modeled, so maybe they could do the they could see the backbone, but they couldn't really make out the positions of the side chain. So the important thing to know is that in the resolution is going to be like an overall value. There can be regions in the crystal that you can't even see, and other regions that you can see, but it's less certain the various position of certain atoms, like side chains of proteins. So like which way, like a lysine or a histidine, like which way it's pointing. Basically, when you look at a structure and you're basically just seeing this stick model, you can't really tell much about the resolution. But now it's integrated. I'm actually going to show you in Primal, but I wanted to show you how you can see here as well. If you go to density enable, and then I'm just going to put, you can do it around focus, or I'm just going to enable it around the whole structure, update. And now you're able to see the electron density map. So if I turn off like this cartoon, you can see that this is actually what you get with the crystal structure. You get something like this, and then you actually have to build the model into it. And you can also see here that you basically, this is a crystal, and so you can see now you have like other units that are coming in that are over here. So you have, this is just showing like a single tetramer, and then these would be like other tetramers that are crystallized alongside it, because this is just a single unit from the entire crystal. But the information from all of them is like the same. It's all combining information, so these would be like identical to this. You can also like display or download files. So if I want to look at the PDB file, so if you, there's like a bunch of information here, but I wanted to show you that for each of the various atoms, you have all of this information about like the atomic positions, and then also things like so this, wait, you can't highlight this this column, but this column is going to be your B factor, and so this is going to be your temperature factor. And so this is going to be that thing we were talking about, like the displacement, or the like kind of how fuzzy a region is. So the higher the B factor, the less certain we are about the position of that various atom. And so you can see that some regions have fairly low B factors, some regions have really high B factors. It's hard to see when it's in this list, but I just wanted to show you where this is coming from. You can learn a lot more about the various, like how to read PDB files, and dealing with coordinates and PDB 101, which I really like. But I'm going to show you in Primal because it is easier. So I'm going to color this a different color. We'll just color this. We'll color this guy teal for now, just so we can kind of make it apart better. Okay, so this is showing basically this, we're seeing it in cartoon view. So we can't even like see the side chains or anything. I'm just gonna stick this, instead of looking at cartoon, if we go to show, we're just going to show it as sticks, and then we're going to hide our cartoon. And we can do this in like the typing in code and stuff, but I just want to do it this way so it's easier for you to see. But anyway, okay, so now we can see where they've modeled the side chains and everything, but this is just where they're saying the side chains are. And so we want to see, okay, well, how did they figure out where the side chains were? How did they figure out the position of all of these atoms? How did they figure out where the model things? And so now let's look at our electron density map. So if I go to action mesh, we'll just do a sigma level of 1. Okay, so here we're looking at like a unit cell, and you can see that you have this one tetramer, and then you can see that you have parts of the other ones coming in as well. And if we, I'm just gonna kind of the focus, so we don't see as much. But if we were to hide the structure, now we're just looking at that map. And so this is what they have to work with when you're doing this. And so you can, when you're building your model, you have something like this that you're then building into. And so we have to keep this in mind when you see a stick structure that, well, they're actually, this is their built in, their model, and we don't know anything about the, like, how high the resolution actually was that they were using to build that model. You could have the same resolution data, and you could build the same model with like a low resolution or a high resolution type of thing, but we really wouldn't be, the low resolution would just be more guesses, whereas with a high resolution, you're more certain. And so with a low resolution, it would be really hard to guess the actual right positions of everything. Whereas with you have a high resolution data, you can more accurately determine, like, yes, this is where this is supposed to go. But just have caution when you're looking at a structure, when you're seeing something that's just sticks, you want to actually go, and if you really care about a region, especially, you want to go and look at the actual maps, the electron density maps, or at least look at the B factors. And so let me show you how we can see those B factors. So if we go to color, we can color, but we can color spectrum, and then B factors. So if we color it by B factor, we can see that the higher B factor, so that's going to be like the redder and oranger things, these are going to be out on the surface typically. And so the things that are more flexible are often on the surface, and they're often like interacting with water and that sort of thing, but they can be more dynamic, whereas things in the center are going to be kind of more tightly held. So you can see that this region on the end, it has kind of like this high B factor. So it's colored in this like dark red. So you can see that we're looking at like the very end of the structure. And you can see that, so for this histidine, like histidine is one of those ones that we can often see like really well on the side chain, make it out really well. But you can see here that you can't, they're kind of they're kind of building it like, there's a bunch of noise that is basically the same as this, so they can't really make out the side chain of this histidine very well, so they have to kind of guess the best location of it. But the point is that if you were to look at a histidine in a region that had a lower B factor, so somewhere where we're looking at like a dark blue, here we can make out the positions of that side chain much better than we could in that region with a low B factor. And so see, we can clearly see the density for this histidine, and we can see the density for this phenylalanine really nicely. And so in these dark blue regions, it's a lot easier to like trace the various positions and be more confident about their locations. Whereas in regions with a high B factor, it's harder to make out the positions of the various atoms. So if I come into Primal, what you can do is just file get PDB. When you do this, so 2BLE, and we want to download this 2FOC map. I'm not going to get into the details about maps. But this, I'm just gonna remove the solvent so we don't see those waters. Okay, display our sequence. There we go. We can see that we have this one chain. So this is just this is just a monomer out of that tetramer. So one of the 4 subunits. We have our GMP, and then we have these sodium atoms, sodium ions. But so let's look at the structure. Okay, so we can see that this, so we can see the structure. So when we're talking about like a television screen or computer screen resolution, typically the resolution there is reported in like the pixel number or density of pixels. So pixels are like separately controllable picture elements that can like display different colors and intensities of light. So if the pixels are smaller than the thing that you want to show, then multiple pixels can kind of work together to show it, but you can't show things smaller than a pixel. There's also something called like a subpixel, which I'm not going to get into. But basically, even if your eyes could tell that there were like 2 dots in one of these squares, if those things are smaller than a pixel, then you wouldn't be able to tell because the pixel would kind of be just like averaging it out. I mean, it could only show you as like a single dot. And so basically the screen resolution is usually reported in like the pixel dimensions. So like common screen resolutions are like 1280 by 720, which is reported as 720p, or 1920 times 1080, which is 180p, 2560 times 1440, which is 1440p. Then you have things like all the way up to like 7680 times 432, which is 8K, or 4320p. Yeah, so basically you can get these crazy numbers, and these like here that's like higher is better. But that's because it's telling you about the number of pixels, and then you can also but if you want to get more detail and stuff, then you have to actually increase the pixel density and not just the number of pixels. And so you're basically packing more pixels into the same area. And yeah, so this is how this is like where you get like the crispness, and then that's reported as like pixels per inch, or PSI. And so yeah, but basically the point with all of that is that the higher numbers are better resolution, because you're talking about like the numbers of the density of the pixels, whereas instead of how fine, like how close together the things that the pixels are actually separating can be, type of thing. And so it's just a difference in convention about how we name these things, where you want a higher number when you're talking about like buying a computer or a TV, whereas you want a lower number if you're talking about looking at a molecular structure.